This conference will now be recorded.
Good morning, I'm Dr. Divya from TeenEinstein. So, in the fifth chapter, Principles of Inheritance and Variation, today we are going to learn the concept of linkage and recombination. What do you mean by linkage and recombination? From the term itself, in genetics, almost everything, you will get a basic idea from that term itself. Recombination means different type of combination, not in the normal expected combination. In different way, it is recombining. Okay. Then linkage. What do you mean by linkage? There is some kind of linkage or there is some kind of relation between genes. So that's what there is some kind of link or linkage between genes, that is called as linkage. And recombination means in different order it is combining. So all this we are going to discuss today. So you know that Mendel selected pea plant as a study material and he conducted his different crosses for all 7 contrasting characters. And he selected based on several reasons all this research material based on the crop duration, then the characters which can be easily identified in each generation. So due to all these reasons, Mendel selected that material. Like that, Morgan also conducted several crosses, but he didn't do that in plant. His material was Drosophila melanogaster fly. In that fly, he conducted his research experiments. So Morgan carried out several dihybrid crosses. In all these dihybrid crosses, he conducted his studies about genes which are sex-linked. Sex-linked genes means in this Drosophila, you can see male and female. Male and female flies you can see in Drosophila melanogaster. But in that, which are sex-linked, in male, which are linked, which are linked to that male gene and which are linked to that female gene. That's what he conducted. For that, he conducted his studies. So he selected and conducted his dihybrid process in Drosophila to study the genes that were sex-linked. So to identify the sex-linked genes, he conducted his studies. So the crosses were almost similar to the dihybrid process, which is carried out by Mendel. Okay. However, how that Mendel carried out his genetics experiment in the pea plant, in the same way Morgan also conducted his experiments. Okay, all this, but it was in plant Mendel conducted, but here Morgan, he conducted his studies in Drosophila, the fly. Okay, so he wanted to find out the sex-linked genes which are related to the male and female sex. So how the genes are related, all this, he concentrated his studies in the sex-linked inheritance of Drosophila melanogaster. So he made the crosses, almost the crossing procedure, everything he made, like the method was same. Dihybrid crosses he did, and it was almost similar to Mendel's research plan. For example, Morgan first he hybridized yellow-bodied. Here you can see yellow-bodied and white-eyed female. So female was here. Female was with yellow body plus white eye. Okay, so females were with yellow body and white eye, and male. how the males were with brown body and red eye. Brown body and red eyes. The females were yellow body and white eye. Here males were brown body with red eye. So he crossed and raised the F1 progeny. First he raised the F1 progeny. He crossed these two and he raised F1 progeny. F1. Then in F1 what they will do? Mendel what he did? Mendel did selfing of F1. So like that, here you know this is a fly. So they can't do selfing because the male one is different and female one is different. So selfing in the sense here in these flies, male is separate and female is separate. So they did intercrossing from the same generation only, not from different. From the among these F1, definitely so many organisms will be there, like so many flies will be there. So among them only they did that crossing, intercrossing in between. So he observed that these two genes, they did not segregate independently of each other. Okay, they did not segregate independently. In which generation? In F2 generation. So after getting that F2 generation, he noticed that there was no segregation, no independent segregation of each other. Okay. So in the F2 ratio, and whatever ratio he got in F2, that was very different, or it was significantly deviated significantly from 9:3:3:1. This 9:3:3:1 is the expected ratio of F2, right? In F2, normally which ratio, according to Mendel's study, he proposed these rules and all. Law of inheritance, they are already having that, right? Up to that, the studies were done. So after that, when Morgan came, he conducted sex-linked studies of this Drosophila. So how he will expect, normally after studying this Mendel's work, how he will expect the result? 9:3:3:1, right? That was the expected ratio when two genes are independent. Okay, but here in F2 generation, in Morgan's study, in F2 generation, that ratio was entirely different, or there was so much of deviation from the normal Mendelian ratio, expected Mendelian ratio. Which ratio he expected? 9:3:3:1. In that ratio, he will get F2 result. That's what he expected, but it was deviated from that ratio. He got a deviated ratio from the F2. Okay. Then Morgan and his group, they knew that the genes were located on the X chromosome. Okay, these genes were located on the X chromosome. So when these two genes in a dihybrid cross, they were situated on the same chromosome. Okay, these two genes. Which two genes? For example, I'll tell you. The sex-linked genes or the dihybrid crosses which we selected, those characters, they were situated on the same chromosome. And the proportion of the parental gene combinations were higher compared to the non-parental type. Okay, compared to non-parental type, the proportion of that combination was higher. So the proportion of parental gene combination was higher compared to non-parental type. And Morgan finally, he made a conclusion that due to the physical association between these genes, due to the physical association or linkage. That is a physical association or linkage between these two genes. Here we explained about two genes, and he found out from his study, he got an idea that all these three genes, maybe A1 and A2, all these two genes, they were located. He made a conclusion that these genes are located on the same chromosome, and there is some kind of physical association between these two genes. Okay, so the genes have some physical association. That's what they called as linkage. So that two genes are linked and they have a linkage or some kind of physical association, and they are on the same chromosome. So he coined the term linkage. Morgan coined the term. The term linkage is coined by Morgan. Okay. Important question, MCQ. He coined the term linkage and he described the physical association. That's what now we have called as, we are calling it as a linkage. Linkage is the physical association of genes on chromosome. Okay. So on a chromosome, suppose two genes are present. These genes, they have some kind of physical association. It is called as linkage. So the term recombination, what is that recombination? It's the generation of non-parental gene combination. Okay. Suppose here, this is parent 1 and this is parent 2. Okay. In next generation, they may get half parent 1 and half parent 2 in that kind of combination. And here, like this. Okay. But other than this, what is recombination? In any way they can combine. So it may come like half, first part like this, then from the another chromosome, when it is during cell division, I'm saying all these combinations. Then again, one more. And here also in the same way, first it may come like this, then this, like this. So in different type of combination you can see that is different kind of recombination. So the recombination, it is the formation or the generation of new parental type. According to this, according to mental studies and all, parental combinations you can see both characters of both the parents. So other than that, some other combinations also may come, that is called as recombination. Okay, any different combination they can find out, or there is a possibility of different kind of combination. So the term linkage, it is coined by Morgan. So the term, what is linkage? Linkage is the physical association of genes on a chromosome. Physical association of genes on a chromosome, that is called as linkage. Okay. And the term recombination, that is the generation of non-parental gene combinations. Non-parental compared to the parents, they have recombinants means they have some non-parental recombination. Non-parental gene combinations. Recombination means it's nothing but other than parental combination, some different combinations. Gene combinations can be seen. That is called as recombination. And here, Morgan and his associates, they found that even when genes were grouped on the same chromosome, some genes are very tightly linked. Okay, there is a tight linkage between some genes, even it is on the same chromosome, they have some kind of tight linkage. Okay, so such tightly linked genes that will not show much recombinations. So there won't be, see, this topic may be a bit confusion. It may create a little bit confusion for you people because in higher classes you will get the clarity. What I'm saying is from one parent, exact parental copy we may get and some slightly different combinations, right? Some combinations you will get like skin color and all we said. Skin color, there are different ranges of skin colors, right? So like that, you may get slight difference in that. But here I'm saying about gene 1. Here, this another gene is gene 2, and next gene 3 is very near to gene 2. This is gene 3. Okay. In such cases, see here, there is a break, or while separation of this chromosome, here it is getting separated. So this portion, see, there is no distance. Distance is, we can't say no distance. Here actually, the distance between these two genes, it is very less. That means whenever this part is getting separated, these two genes, genes 2 and 3 may get separated. When it is coming in between here, between 2 and 3, then only the chances of separation is coming. Otherwise, almost every time while breaking, see 1, or while cell division, 2, this is 2, this is 1, and this here it's coming near a third gene. Okay. So here, whenever this is, or the chromosome, it is getting separated. So while getting separated, this here there is a chance of breaking, right? So this chance is more because this distance between gene 1 and gene 2, it's more. But the distance between gene 2 and gene 3, it is very less. So here almost very rare chances of breakage here. Every time it won't get the same breakage. But here, you in any point, here points are more, right? So at any point, they will get a separation. So whenever that distance is less, there is very less chance of recombination. Distance between genes are less means there is low chance of recombination. See here. Here one, these are the genes, yellow, white and all. So if this distance is more, you may get more number of recombination so that the genes may go in different combinations. Okay. But when that distance between genes are less, you will get very less recombinance. And other genes, if they are loosely linked, that shows high recombination. Okay, so you may get problems related to this concept. That time you should notice, like while reading the question itself, you should note down somewhere where which gene is very near, which gene is far, the distance they will mention. While doing the problems, it will be very easy. First, you should mark on a chromosome. This is the A gene, gene A and gene B, the distance between these two genes, or they are very near. But gene C is far. So recombination in between A and B, it will be very less. The frequency of recombination should be very less according to this rule of recombination. Okay, so it's just like a general thought you can make out very easily by mentioning the distance. So from the B gene and C gene, there is a gap or distance is more. Okay, genetic distance is more between those two. So how you can make out? It is very easy. So there between B and C, recombination chances or recombination frequency is more. But in the case of nearby genes, the frequency is very low. So here white and yellow, where here this white and yellow, in this you can see this is yellow and this is white. They are very tightly linked. Here the distance is very less, tightly linked, and 1.3%. The recombination is 1.3%. Okay, white and yellow it is showing. And Morgan's student Alfred Stewart, he used that frequency recombination between gene pairs on the same chromosome. Okay, so that they can consider as a measurement for the distance between genes. Okay, Alfred Morgan's student, okay, he mentioned this distance as a measure of recombination. So if that distance is less, here we have marked it as 1.3%, the recombination frequency, because it is very near. The distance is very less. So recombination frequency also will be very less. If the genes are far, then there is more chance. See here, wild type. Here the distance is more. Here recombination chances or recombination frequencies will be more. Here you can see this yellow and white. Here recombination you will get only 1.3% because however it is combining also these genes will get inherited together. So there is less chance of recombinations. Okay. But in the case of this wild here 37 recombinants 37.2%. You can see parental type 62. Here parental type 97. Where the genes are near, there or the distance is less, their parental type 98% and recombinants only 1.3%. But if the distance between genes are more, parental type will be 6. Here in this case, parental type was 62.8 and recombinants they got 37.2. That means the percentage is getting increased in the case of more distance. Okay, so this distance they consider as the measurement for the frequency of recombination. Next, we need to say about theory of this linkage. Okay. So in theory of linkage, what they are saying, the linked genes, they are linearly located on some chromosome and they get separated if exchange or crossing over takes place between them. Okay. So these linked genes are linearly located on chromosome. Just now I showed you. This is chromosome. Think. Assume this is chromosome. Gene A and gene B are linked. Okay. Then these genes are linearly located. If it is not linearly located at the stage of cell division, they won't get inherited together, right? So they are linearly located on the same chromosome and they will get separated. How they will get separated? If exchange takes place between them. Here, in this point, exchange taking place, then only it will get separated. So the linked genes, they are linearly located on the same chromosome and they get separated if exchange is taking place between them. Okay. And next, linkage group. So all the genes which are located on one pair of homologous chromosome form one linkage group. Okay. Which are located on one pair of homologous chromosome. One pair of homologous chromosome, those genes are called as linkage group. So all genes which are located on one pair of homologous chromosome, they form one linkage group. Okay. So the genes which are located on homologous chromosome, they will get these genes will get inherited together. So we can consider it as a linkage group. Here, see, these are linkage groups. Homologous chromosome, they will get inherited together. So they are called as linkage group. So the number of linkage group, how can you find out the number of linkage group? Number of linkage group, it is equal to number of homologous chromosome pairs or haploid number of chromosomes. How you can find out the number of linkage group? Number of linkage group. How you can find out? Number of linkage group, it is equal to number of homologous chromosome pairs or haploid number of chromosomes. Okay. So in pea plant, the diploid chromosome number 2n is equal to 14. So how the haploid number will come? Haploid means n number of chromosomes. So 14, n is equal to 14 divided by 2. So it will come 7. Okay. So that is having 7 pairs. 7 pairs of chromosomes. So the linkage group, how many linkage groups you can expect? 7 linkage group. In the same way, in Drosophila, diploid chromosome number. Drosophila, diploid chromosome number is 8. Like this, some important organisms or important here plants, study materials, chromosome number you should remember. In Drosophila, 2n, diploid number of chromosome it is 8. See, this is the almost the main reason for selection of this material, Drosophila, because chromosome number is very less. So they will get very less of, they will get more number of characters, and the study will be very easy. If they have 2n equal to, in human beings, and it is very difficult to study because they have lot of genes and lot of combinations will come, right? So it will be very difficult. So study material should be the common one, where we can identify the characters very easily. Okay. So the Drosophila here, diploid number of chromosome is 8 and haploid number n will be 4, right? So how many pairs? 4 pairs. So how many linkage groups? 4 linkage groups. Okay. So the number of linkage group, it is equal to number of homologous chromosome pairs or haploid number of chromosomes. And if the distance between two genes is increased, then the strength of linkage is reduced, and it provides that greater is the distance between genes, the greater is the probability of their crossing over. Okay. So if the distance is less, they will get inherited together, and if the distance is more, there is a chance of separation. The chance of separation of these genes are more. Okay. So the distance between two genes increased means then the strength of the linkage, it will get reduced. The strength of a linkage will get reduced, and there a greater chance to get separated. Okay. When it is near, there will be very less chance to get separated. These genes won't get separated. In all cell division, there is a minimum percentage of separation only. Okay. So the distance between two genes is increased, then the strength of the linkage is reduced. If distance is increasing, distance increasing means linkage percentage decreases. Okay. The next arrangement of next we are going to learn about arrangement of linked genes on chromosome. See, these topics are not there in your textbook. And for your board exam, these topics are not important. But for NEET exam, these topics are very important. This is not there in your textbook. So note down arrangement of linked genes on chromosome. So arrangement of this linked genes in any dihybrid, it is 2 types. In any dihybrid, you will get 2 types of arrangements of genes. Okay. First one is cis arrangement. First one is cis arrangement. What is cis arrangement? In this, 2 dominant genes are located on one chromosome and both recessive genes are located on another chromosome. Okay. There is cis arrangement and trans arrangement. There are two types of arrangement of genes on a chromosome, cis arrangement and trans arrangement. So what is cis arrangement? The dominant genes, they are located on one chromosome. See here, in one pair of chromosome, here dominant genes. Here one gene is present, here one gene is present. Gene A and B, they are present in one chromosome. Dominant genes are present in one chromosome, and another allele, the second one, the recessive allele will be present on another chromosome. Okay. So the dominant genes are located on one chromosome, and both the recessive allele or the recessive gene, it is located on another chromosome. So such type of arrangement, that is called as cis arrangement. This is called as cis arrangement. So the dominant genes will be present on one chromosome, and the recessive one will be present on the another chromosome. Okay. Don't get confused. This is not on separate like chromosome. It will be on separate chromosomes, but with of the same pair. Okay. Chromosomes will be in pairs, right? In first one in that pair, on first chromosome, the dominant alleles will be dominant genes will be present, and another one recessive genes will be present. It won't come like one dominant and one recessive, one dominant and one recessive. That's what they are saying. So the two types of gametes can be produced in cis arrangement. Here, two types of gametes will come in cis arrangement. Which are the two gametes? Here, from this one gamete, which is the gamete gamete AB, and from this another gamete AB. Okay, there are two types of gametes. Gamete, this is dominant with dominant genes, and this gamete is with recessive genes. Okay, this is called as cis arrangement. So there are two types of arrangement of linked genes on a chromosome. How they will get arranged? There are two types of arrangement, cis arrangement and trans arrangement. In cis arrangement, two dominant genes are located on one chromosome, and both recessive genes are located on another chromosome. This type of arrangement is termed as cis arrangement, and cis arrangement, that is the original arrangement. Okay, cis arrangement is the original arrangement, and there are two types of gametes in cis arrangement. First one is with both dominant one, and another one is with both recessive genes. So this is one gamete. You can see here. This is one gamete and this is another gamete. Okay. Next type is trans arrangement. Here you can see the trans arrangement. What is trans arrangement? In this, a chromosome bears one dominant and one recessive gene. Okay. Here, suppose this is one chromosome and this is the another pair chromosome. Here, first gene A, and in the previous one, in one chromosome, both were same. Means both were dominant, and in that homologous one, both were recessive. But in this case, one dominant and one recessive. And here, the second one will be dominant and the another one will be recessive. So here, in this case, the chromosome bears one dominant and one recessive gene, and another chromosome, on another chromosome also one dominant and one recessive gene. So this type of arrangement is called as trans arrangement. What is this? Trans arrangement. cis arrangement and trans arrangement. So this trans arrangement is not the original form. Okay, trans arrangement is not the original form. Cis arrangement is the original form. And how the trans arrangement is forming? It is due to crossing over. The normal one is the cis arrangement. How trans arrangement will come? It is due to crossing over. You got that what I'm saying? See, you will get, I'll tell you, like this chromosomes. Here, A and B. We said this is the original one, right? This is A and this is B. This is the normal arrangement we said. Normal arrangement of genes on a chromosome, right? So, that is the original form. Here, the trans arrangement is not the original form and it is arising due to crossing over, like that. These chromosomes will cross over. See, this may form like, this will cross. It will align in this form. Okay. So, while crossing over or the crossing over time, one half like this and like this, it will form. So again, after cell division, how it will form? This will become a pair, small a and capital B. And next will be like this. This is the crossing over. It is crossing over another chromosome. So finally, the pair will become here, this part, B. And the second half will be from here. Okay. First half will be like this and second half from here it will come. Then it will, that here in the another half they will get the recessive gene. And here, sorry, this should be small, right? Here, B and see like this, B and capital, this part up to here B, then this part is A. So it is forming a cross. This is the crossing over and this trans arrangement is due to the crossing over. This is not the original form. It is due to crossing over. So two types of gametes also formed in trans arrangement, but it is different from cis arrangement. Okay. In cis, you will get AB and AB gametes, right? But in trans, you will get AB and AB. Here also two types of gamete, but the combination style is different. Okay. So cis arrangement and trans arrangement, two types of arrangement is there. There are two types of linkage. Types of linkage we need to study. What is that? Types of linkage, that physical contact or physical combination, that different combination. There is a, if the distance is increasing, linkage will decrease. Or the distance is decreasing, that linkage will increase, right? So that there are two types of linkage. So complete linkage and incomplete linkage. What is complete linkage? The linkage in which genes always show parental combination. That is called as complete linkage. So it never forms a new combination. Parental combination only it will form. Okay. There won't be any new combinations. Only parental combination it will form. That is the complete linkage. Linkage in which genes always show parental combination and it never forms a new combination. That is called as complete linkage. Okay. Those genes will be always inherited together. So there is no chance for a different combination. That means that genes, the linkage distance between those genes are very less. So they'll be getting inherited together, all those genes. So that is called complete linkage. And in such cases, crossing over is absent. These terms you should note down, complete linkage, because when you are getting a question, they'll mention us there is a complete linkage. So you should know what do you mean by that term, complete linkage. Means the crossing over is absent in it. So such genes are located very close on the chromosome. So the genes will be very nearby. Okay, that distance will be very less. So such type of linkage is very rare in nature. That is very rare in nature. In male Drosophila, then female silk moth and all you can see. It's very less. Number of such linkage, it is very less. So you can see such type of linkages in male Drosophila, then female silk moth. So that there is no chance of crossing over or recombinations are very less in that. There is no new combination. And what is incomplete linkage? What do you mean by incomplete linkage? In incomplete linkage, these new combinations also appear with parental combination. There not only the parental combination, there are new combinations. New combinations also will be present in the incomplete linkage. When the linkage is complete, the genes will be located nearby and there is less chance of crossing over and there won't be new combinations. In very less cases we can see such complete linkage. In male Drosophila you can see, then female silk moth you can see complete linkage. Then there only parental combinations you can see. Then in incomplete linkage, new combinations also appear with parental combination. Along with parental combination, new combinations are also present. So these new combinations are due to crossing over. Just now I explained what is crossing over. So due to that crossing over, new combinations also, other than parental combination, new combinations also will be available. Okay. Then genetic map or linkage map or chromosome map. What is this genetic map or linkage map or chromosome map? In genetic map, different genes are linearly arranged according to percentage of recombination between them. The genetic map, in that map, this here. In that genetic map, different genes here, gene A, gene B, gene C, gene D, gene E, here, gene F, gene G, like that several genes will be there. Okay. And here also A. Okay. So such genes will be marked on the chromosome. This is a whole genome. In that everything will be marked. So in genetic map, different genes are linearly arranged according to the percentage of recombination between them. Okay, that is the each chromosome. In each chromosome they will mark. So with the help of this genetic map, we can find out the position of a particular gene. We can find out it. With the help of that genetic map, we can make out the position of a particular gene on a chromosome. On a chromosome, where is the position of that particular gene, that we can make out with the help of this genetic map. So if the gene distance between genes are more, there is a very less chance of linkage. But here, see here distance is more. But in this case, the distance is less. So here, that linkage will be more. Chances or the frequency will be more. But here it will be less compared to that one. Okay. Very nearby genes, they will get inherited in the next generation, or that frequency will be more. Always they'll be getting inherited. Okay. So with the help of this genetic map, we can find out the position of a particular gene on a chromosome. On that chromosome where this gene is located, that we can find out. And the genetic map, it is helpful in the study of genome. For genome studies, this genetic map is helpful. Okay. The genetic map is helpful for the genome studies also. These points are not there in your textbook. And this is not mainly for board exam. This is for NEET exam. These points: genetic map, linkage, recombination, crossing over. You should know all these things. It's mainly for your NEET exam. Then polygenic inheritance. You know, Mendel studied mainly the characters or the traits. They have distinct forms or they have alternative forms. Like, for example, flower color. Mendel selected such characters only where they have a contrasting character, like purple flower and white flower. Exactly, we can identify very easily. It is exactly different from one character or the trait. There is a different form of that same gene. Okay, but if you are seeing in nature, we can find out many traits. Okay, many traits which are not so distinct in their occurrence, and you can see such types. So many types you can see. Like this, you can't exactly differentiate. So in so many cases are there like that. In humans, we have already explained. In humans, tall or short people we can't say. In plant, in the case of Mendel's experiment, he was able to identify tall plant and dwarf plant. But in the case of human being, how we can say that it should be? We cannot say like that because there is a range of that height, right? We can't expect in both extremes. Always we can't expect in both extremes. If extreme means how it will be, all will be tall or they'll be short or they'll be dwarf. How we can expect human beings height like that? We cannot. So there is a wide range of that height, right? So human, we can't expect like tall or short people. So two distinct alternative forms, but there is a range for that possible height. Okay, medium height people will be there. See, one people will be 180 centimeters, they'll tell. And some people will tell 172, some may tell 150. In female, they will tell 160, 150, like that. So there is different range for normal height, right? So traits that are generally controlled by 3 or more genes, they are called as polygenic. Poly means more, multiple. So the polygenic traits. Polygenic trait means the gene, the character which are controlled by 3 or more genes. The character which are controlled by 3 or more genes, they are called as polygenic trait. So beside the for the involvement of this multiple gene, polygenic inheritance that also takes into account of the influence of environment. So there is an effect of environment, not only the gene, genetic effect, gene plus environment, both makes the final result, right? For example, if you're planting a plant in Bangalore, the climate is different or the environmental condition is different. The same thing you're planting in Tamil Nadu or Chennai, the climate is different. So the growth, they will give the result, an average result, both will give average result. But for that plant, which is the suitable condition or most acceptable condition, in that it will produce or it will give more result, right? So like that, there is a connection between the gene and the influence of environment in all traits, not only this one, almost all traits. So the multiple genes, not only that multiple genes are involved in the inheritance of character, there is an effect of environment or there is an influence of environment in the expression of those traits. Okay. Then, for example, we are saying human skin color. In human skin color, it is another classic example for polygenic inheritance. So in a polygenic trait, that phenotype that reflects the contribution of each allele. So effect of each allele is additive. There is an additive effect. See, gene A, B, C means this together it will make an expression. So there is an additive effect. We have already discussed about this polygenic inheritance. So just think that there are three genes A, B, and C. These three genes controls the skin color in human. Okay, when in human with the dominant forms, in dominant condition, dominant A, B, and C. That when all these three genes are coming in dominant condition, that forms the dark skin color. Okay, and when it is coming in recessive condition, it is recessive A, recessive B, and recessive C, that forms light skin color. When all three genes are coming in dominant condition, that will form the dark skin color, and when it is coming in lighter version, it will be in recessive condition. All these three genes will be recessive condition. So the genotype with the dominant allele, they have the darkest skin color, and all recessive allele they are coming means they have the lightest skin color. Okay. So in a genotype with 3 dominant alleles and 3 recessive alleles, like in this combination it is coming means they will form intermediate skin color. If it is all dominant homozygous condition, that forms the darkest skin color, and all recessive homozygous condition, that forms the lightest skin color. So intermediate means one dominant allele and one recessive, one dominant, one recessive, one dominant, one recessive for all three genes. This forms the intermediate skin color. So in this way, the number of each type of allele in one genotype, that determines the darkness or lightness of the skin color in one individual. Okay, in human beings, skin color, it is controlled by 3 genes. If the genes are in dominant, homozygous dominant condition, that forms the darkest skin color, and when they are in homozygous recessive condition, that forms the lightest skin color. Okay, so an intermediate, each gene, one, if it is in a heterozygous condition, that means here you can see one dominant and one recessive, one dominant and one recessive, one dominant and one recessive. This forms an intermediate skin color. So the alleles, number of alleles and the type of that allele, that determines the darkness or lightness of the skin color of individual. Got it? Then next is pleiotropy. What is pleiotropy? The effect of a gene on a single phenotype or trait. Okay. Effect of a gene on a single phenotype. On a single phenotype, the effect of a gene, that is called as pleiotropy. So how we can explain this? If there is a single gene that can exhibit multiple phenotypic expression, like, so that type of a gene, that is called as pleiotropic gene. So the mechanism of this pleiotropy in most cases, it is effect of a gene on metabolic pathways, and this contribute towards different phenotypes. So there is a effect of a gene on the metabolic pathway, and that effect, that metabolic pathway effect, or the effect of that particular gene on the metabolic pathway, that contributes different type of phenotypes. So an example of this is phenylketonuria. Phenylketonuria. So that occurs in humans. In human beings, phenylketonuria, that is a disease, phenylketonuria disease, which is present in humans. So the disease is caused by the mutation in the gene. So there is a gene mutation. Because of that, this disease is coming, and that codes for enzyme phenylalanine. This enzyme phenylketonuria, that is due to the mutation in one gene, and that gene codes for enzyme phenylalanine hydroxylase. This is the enzyme name. And this is a single gene mutation. Gene mutation and all you can study in the next chapter. So the single gene mutation, one gene is getting mutated and that is creating metabolic different effect on metabolic pathway, and that gives a different phenotype. Okay. So what is pleiotropy? Effect of a gene on a single phenotype. One gene is affecting the phenotype, one single phenotype, and that is due to the that gene is affecting on the metabolic pathway. That particular metabolic pathway, if it is going in a normal way, it will produce a particular phenotype. And if there is any change or any mutation in that change, that will create an effect on the metabolic pathway, and finally it will result in a different phenotype. So example of this pleiotropic gene is in human being one disease like phenylketonuria, and this disease is caused by the mutation of a single gene, and this gene codes for the enzyme, which is that enzyme phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzyme. So that is due to a single gene mutation. It is due to a single gene mutation. Is it clear up to this? Is it clear, or you want me to repeat anything? Deepa, do you have any doubt, or you want me to repeat?
No, ma'am.
It's clear?
Yes, ma'am.
Okay. Then sex determination and sex-linked inheritance and all we will study in the next class. Sex determination in human beings and all. So that's all about today's class. Thank you.